Welcome to Tomorrow Unveiled.
We're diving deep into the engine room of the future, specifically focusing on the most rigorous, cross-validated scientific breakthroughs in longevity science.
And this deep dive covers the pivotal week of November 11th through the 18th, 2025.
That's right.
Our mission today, as always on Tomorrow Unveiled, is to be your high-speed filter. We've gone through a huge stack of high-level sources, research articles, expert notes, all compiled by our dedicated research team.
And we're pulling out the confirmed reality from, the pure hype.
We're looking for the breakthroughs that are actually moving the needle in human health span.
And that focus it has never been more critical, especially when you look at the big picture context of aging.
Right.
The information our research team put together, it really underscores a sobering reality that provides the entire rationale for this field.
So tell us about that reality, because we're constantly being told that we're living longer year after year.
Historically, that's true, but the rate of progress is slowing down.
Yeah.
Dramatically.
Stalling out.
The sources we reviewed highlighted these recent demographic analyses, and they show that the traditional gains in life expectancy we've all relied on for a century, they're hitting a wall.
We've picked the low-hanging fruit.
Exactly. We did great with public health, tackling early life mortality, but we've really stalled when it comes to late life decline.
So if you were born today, you're not automatically guaranteed to hit 100, even with all of our modern medicine.
That is the key implication. The data suggests that without a major systemic intervention that fundamentally targets the aging process itself, no generation born after 1939 is expected to reach an average lifespan of 100 years.
Wow.
It basically confirms that the conventional medical model, wait for heart failure, diabetes, or Alzheimer's to show up and then treat them one by one.
That model has hit a point of diminishing returns.
Exactly. It's like the difference between preventative maintenance on your car and just, waiting for an emergency breakdown on the highway. We've been focusing on the crises.
And that shift, that brings us to the core idea driving all of this modern research, right? The Geroscience Hypothesis.
Yes. The Geroscience Hypothesis. It's this understanding that these major chronic diseases are not independent problems.
They're all connected.
They share the same handful of fundamental biological drivers, the hallmarks of aging. And if we target that underlying process of aging itself, we can delay or even prevent the onset of all those diseases at the same time.
So you're fixing the master clock, not just replacing the gears one by one as they break down.
That's the perfect analogy. You're reframing the entire medical mission.
So what's the confirmed roadmap for that mission this week on Tomorrow Unveiled?
We've broken this down into 3 major areas that showed corroborated progress. First, we're going to look at new chemical interventions, specifically how things like senolytics and metabolic regulators are being combined for synergy.
What?
Second, we're looking at this astonishing leap in, engineering the human blueprint. We're covering precision mitochondrial DNA repair and some really incredible projections for age reversal.
And finally,
the indispensable role of the measurement revolution. This is where artificial intelligence and highly validated biomarkers are just accelerating everything.
All right, let's start with the chemical interventions. That feels like the most immediate, the most translatable area for human health. And the biggest buzzword right now seems to be senolytics.
Zombie cell clearers, yeah.
We call them that, but let's get a bit technical for our listeners. What is cellular senescence, and why is clearing those cells so essential?
Okay, so cellular senescence is one of the most visible hallmarks of aging. Essentially, a senescent cell is one that has detected some kind of damage, maybe to its DNA, or it's replicated too many times, and it slams on the brakes.
It just stops.
It enters this irreversible state where it can't divide anymore, which is good. You don't want damaged cells replicating. But the key is it also refuses to die through programmed cell death, or apoptosis.
So they're not dividing, but they're not dying either. They are quite literally biological zombies just taking up space.
Correct. But the problem isn't just that they're dead weight, it's what they're actively doing while they sit there. They secrete this really potent and destructive cocktail of molecules. It's called the senescence-associated secretory phenotype, or SASP.
SASP. And this is a mix of what, inflammatory signals?
Pro-inflammatory cytokines, growth factors, proteases. It's just a nasty mix.
And this SASP is the source of inflammaging, right? That low-grade chronic inflammation that seems to be underneath everything from heart disease to joint pain.
Precisely. It's inflammatory noise that just disrupts the function of all the healthy tissue nearby. And senolytics are drugs designed to exploit the very survival pathways those zombie cells use to avoid death. They kill them selectively without harming the healthy cells.
So you eliminate the zombie cells, you silence the SASP.
And you quiet down the inflammaging.
Okay, so let's move from the theory to the actual human data. Our research team highlighted a really encouraging pilot study this week on a specific senolytic combination, dasatinib and quercetin, or D plus Q.
D plus Q, yeah.
And this was in older adults at risk for Alzheimer's disease. Given how hard it's been to find effective Alzheimer's treatments, how significant is this preliminary human data?
Oh, it's extremely significant. And not just for Alzheimer's, but for validating the whole Geroscience hypothesis itself. I mean, if senescent cells drive neurodegeneration through inflammation,
then clearing them should, in theory, improve brain function.
Exactly. Now, the pilot study was mainly about safety, and it confirmed D plus Q was well tolerated in this, vulnerable, older population. But it's the secondary data points that really got the scientific community's attention.
That was the potential improvement in cognitive function, wasn't it?
Yes. And while it was a small study, you have to remember, they saw measurable improvements in cognitive scores. And here's the crucial link.
Go on.
The improvements were strongest in the patients who had the lowest cognitive baseline scores to begin with.
So the sicker the patient was, in a sense, the bigger the potential benefit they saw from clearing out all that junk.
That's the interpretation, yes. But the real breakthrough was the correlation with the biomarkers. They were able to draw a direct line. The researchers measured a drop in a key inflammatory marker, TNF-alpha.
Which is a big part of that destructive SASP cocktail you mentioned.
A very high-profile part. And they found that the amount of TNF-alpha reduction in a person's body was significantly correlated with how much their cognitive scores went up.
Now that is the gold standard of mechanistic proof, isn't it? You're linking the drug's intended action-reducing inflammation directly to the functional outcome better cognition in human beings.
It's powerful initial evidence that senescence is a targetable mechanism in human neurodegeneration. But this actually brings us to an important nuance in the terminology we should clarify.
Okay.
The difference between senolytics and senomorphics.
Right, we mentioned this. So D plus Q is a senolytic, the killer. What's a senomorphic?
A senomorphic is a compound that doesn't necessarily kill the senescent cell. Instead, it modulates or silences the SISP.
It stops the zombie cell from shouting out all those inflammatory signals.
You got it. Think of a senolytic as executing the zombie and a senomorphic as cutting its vocal cords.
And why is that distinction so important when we start talking about common longevity compounds, things like metformin and rapamycin?
Because metformin and rapamycin are classic examples of senomorphics. They don't typically cause that acute, widespread clearance that you see with D plus Q. Instead, they regulate core metabolic pathways like MTOR, which ultimately just dampens the inflammatory output of senescent cells.
So it's a dual approach, killing and silencing.
And that's where the field is heading, which leads us right into our next key finding on combination therapies.
Okay, let's talk about those metabolic pathways. Rapamycin is, well, it's maybe the single most potent chemical longevity intervention we've ever found, largely because it inhibits MTOR.
The mammalian target of rapamycin.
What is MTOR, and why does inhibiting it seem to tap into some kind of fundamental aging clock?
So MTOR is essentially the cell's master regulator for growth and nutrient sensing. When nutrients are plentiful, like you've just eaten a high-protein meal, MTOR gets activated, it signals the cell to grow, to divide, to build new proteins.
It's the accelerator pedal.
It's the accelerator pedal. And when nutrients are scarce, say during fasting or caloric restriction, MTOR activity drops dramatically.
So rapamycin is basically mimicking fasting by stepping on the brakes of that accelerator.
Precisely. And our research team reviewed a meta-analysis that, well, it conclusively affirmed rapamycin's efficacy. It extends lifespan almost as consistently as strict, really arduous dietary restriction. And that's across a huge range of vertebrate species.
Which is incredibly compelling evidence.
It is. It's seen as a way to get the biological benefits of caloric restriction without having to be hungry all the time.
But as you hinted at, there's always a trade-off. You can't just slam the brakes on the system indefinitely without consequences.
Right. The big catch, the thing that has really hindered rapamycin's progress to widespread use, is this well-documented side effect of glucose intolerance.
Right.
Since MTOR is so deeply involved in nutrient sensing, blocking it chronically starts to interfere with insulin signaling. And over time, many subjects on rapamycin develop a sort of diabetic-like state where their body struggles to process sugar.
That seems like a massive obstacle. You don't want to trade a late-life disease for a midlife metabolic disorder.
Yeah.
How is the scientific community getting around that?
This is where the synergy becomes the breakthrough. The key data we reviewed this week is all about combination therapies. Researchers realized, well, why not use a known metabolic corrective to fix the side effect caused by the longevity drug?
And that's where metformin comes in.
That's exactly it. The key finding was that co-treatment with metformin was shown to reliably alleviate rapamycin's metabolic problems.
Metformin, the classic affordable xenomorphic we just talked about, it's been used safely for decades to treat type 2 diabetes.
The very same. In mouse models, and the data here is particularly strong in female mice, combining rapamycin with metformin successfully uncoupled the longevity benefits from the metabolic harm.
So rapamycin still extended life.
But metformin blocked the progression of glucose intolerance that rapamycin was causing over time.
So metformin is effectively the safety switch that lets you get the maximum benefit from rapamycin. This is huge for clinical translation, isn't it?
It is.
It means we might be able to use the most effective longevity compound in humans safely as long as we pair it with the right chaperone drug.
It highlights a major theme in geroscience, which is that these compounds are probably not going to be used alone. Longevity will be a carefully curated cocktail.
Yeah.
The combination therapy manages the side effect, which makes the whole package viable.
What's so interesting, though, is the contrast you mentioned about metformin when it's used by itself. It's maybe the most talked about longevity drug, yet that meta-analysis suggested it showed no clear lifespan benefit on its own.
Right.
So if it doesn't directly extend life, why is it so essential in these combination trials?
The distinction is really vital. Metformin is phenomenal at correcting age-related metabolic dysfunction and reducing that inflammatory output. It's a powerful health span booster and a senomorphic.
Yeah.
It prevents a lot of the secondary complications of aging, which is why it's the focus of the famous TAME trial, testing it against the incidence of age-related disease.
Right.
However, the cross-species data suggests that in terms of hitting that core systemic mechanism to dramatically extend overall lifespan, that acute break on the growth pathway, rapamycin, the MTOR inhibitor, is the primary driver.
So metformin's role is supportive.
It's supportive, it optimizes the environment, reduces inflammation, and critically, it neutralizes the side effects of stronger interventions.
So the future is the symphony, not the solo act. One drug to drive the primary effect and another to manage all the downstream complexity.
That's the perfect summation. This combination data is a massive step forward for pharmaceutical aging intervention. It moves us much closer to clinically viable, well-tolerated drug cocktails.
If chemical interventions are about regulating the cell's environment, our next section moves into actual engineering. We're talking about fixing the biological code itself, starting with the cellular power grid. The mitochondria.
And mitochondrial dysfunction is one of the most accepted and debilitating hallmarks of aging. When the mitochondria, which generate 90% of our cellular energy,
ATP,
yeah, when they start failing, every high-demand system, the brain, muscles, the immune system, it all suffers. You just can't achieve functional health span extension without addressing the mitochondria.
The primary challenge here has always been technological, right? Mitochondria have their own distinct circular DNA, mDNA, and our standard gene editing tools like CRISPR just can't get in there.
That's right.
Why is mDNA so hard to edit?
It's a combination of physical barriers and its unique structure. The mDNA is inside the double membrane of the mitochondria. Traditional CRISPR systems are these large protein complexes, just way too bulky to get across those membranes efficiently and stay active.
So this physical bottleneck meant that mDNA repair was basically a pipe dream for a long time.
It was, but this week, the sources our research team compiled confirmed a major technological breakthrough that seems to have solved this bottleneck.
Okay, tell us what happened.
We saw the successful deployment of these novel gene editing tools that are capable of correcting disease-causing mutations in mDNA, and this was demonstrated inside primary human cells.
How?
They used a specially designed system. It's a double-stranded DNA diminase toxin, A-derived cytosine base editor, or DDCBE, and it's guided by tail structures.
Okay, that's a mouthful. Let's unpack that jargon. So instead of trying to force a huge piece of machinery into the mitochondria, what does this DDCB tail system actually do?
Think of it this way. Traditional CRISPR acts like a molecular scissor. It cuts double-stranded DNA. That's complicated to get inside. The DDCBE is a form of base editor.
A base editor.
It doesn't cut the DNA. It chemically changes one base letter, say a cytosine C, into another, like a thymine T. It's a much more precise, chemically nimble process.
So it's more like a molecular pen than a molecular scissor.
That's a perfect analogy. And the tail guides. They act as the highly specific delivery mechanism. They allow the editor to be efficiently imported through the mitochondrial membranes to the exact spot on the mDNA where the correction needs to happen.
So that removes the size problem and lets you do high-precision single-letter edits.
To the circular mDNA, yes.
This sounds like a complete game changer. What's the immediate significance of this for longevity science?
It's profound. I mean, it provides a direct necessary pathway to address that decline in mitochondrial function. If aging is characterized by an accumulation of damaged mDNA that's slowing down energy production, this technology lets scientists go in and essentially refresh the mitochondrial energy grid.
It's a foundational complement to all the other strategies.
It is. You need energy to clear senescent cells, to maintain epigenetic health. You need energy for everything.
Well, we have to talk about the complexity here because mitochondrial genetics is different from nuclear genetics. The challenge is called heteroplasmy, right?
Correct. And this is the next critical challenge. You know, when we talk about our nuclear genome, we have 2 copies. But with mDNA, a single cell can contain hundreds, sometimes thousands of copies of the mitochondrial genome.
And heteroplasmy is the mix of healthy and mutated copies inside that one cell.
Right.
So if you edit 1 or 2 mDNA copies successfully, but 998 others are still damaged, the cell isn't going to function any better.
Precisely. To effectively treat a disease or reverse age-related decline, the correction doesn't just need to happen. It needs to achieve a high and stable correction efficiency across the majority of those mDNA copies.
And how is this new technology doing on that front?
The new DDCBE technology shows really strong promise in patient-derived cells, but optimizing the delivery and ensuring that every cell in a tissue gets that high percentage of correction. That remains the complex, but now, conquerable challenge for the field.
If you're finding this insightful, please take a moment to like this video, subscribe to Tomorrow Unveiled, and consider hitting that new hype button to help us reach more viewers. And of course, be sure to turn on notifications so you never miss an update on the future.
So if mDNA editing is fixing the power plant, then epigenetic reprogramming is the ambition to reboot the entire operating system of the cell.
Wiping the slate clean and achieving genuine age reversal. This is the field that gets massive investment and, frankly, intense debate.
And it should. It represents the ultimate goal, not just slowing aging, but actually reversing cellular time. This field uses molecular reprogramming, often involving the partial activation of the 4 transcription factors first identified by Shinya Yamanaka.
Yamanaka factors.
These factors are essentially the master keys that can force an adult differentiated cell to revert all the way back to a pluripotent stem cell state to become young again.
But the major historical hurdle, as I recall, was that turning an adult cell back into a stem cell is inherently risky. Stem cells can divide uncontrollably and form tumors. You were basically trading aging for cancer risk.
That was the initial massive safety hurdle. The breakthrough in recent years, the one that justifies these aggressive projections we're seeing now, is the concept of partial reprogramming.
Partial reprogramming.
Instead of activating the Yamanaka factors long enough to fully revert the cell to that high-risk state, scientists are activating them only briefly, just long enough to initiate an epigenetic reset.
So you start the reset sequence, but you stop it before the really dangerous transformation occurs. You get the benefits of youth without the full-blown developmental risk.
That is the key. And the projections coming from experts who are deep in this field, they are truly eye-opening. Our sources shared projections suggesting that through this modulated reprogramming, tissues could potentially be rejuvenated by 50%, 75%, or even 90% of their biological age.
90%? That is an astonishing claim. What would that even look like functionally? Taking a 70-year-old organ and making it act like it's 7?
Functionally, it means vastly improved tissue resilience, regenerative capacity, functional output. If these technologies prove safe, they represent a path to eradicating many age-related functional deficits all at once. But the most shocking part of this is the projected timeline for human translation.
The timeline shock. What are the experts projecting?
The consensus projection that our research team noted is that age-reversing pharmacological pills capable of safely inducing these partial epigenetic resets, they could potentially be ready for human use by approximately 2035.
2035, that's barely a decade from now. That implies an enormous amount of safety and optimization work is happening as we speak.
It is. The primary focus right now is ensuring precise temporal control over the factor expression. The pill can't be a perpetual youth cocktail, right?
Right, yeah.
It has to induce a temporary controlled pulse of rejuvenation, and then the factors have to be fully deactivated to prevent any risk of teratoma formation or malignancy.
And what about delivery?
That's the second hurdle, developing a stable, orally administered compound that can reach target tissues efficiently and safely. That's a huge pharmaceutical challenge, but one they believe can be solved within the next 10 to 12 years.
And that transition from complex gene therapy to a simple pill,
Yeah.
that's why the timeline is so aggressive.
It's exactly why. If we just zoom out for a second, the combination of mDNA repair and epigenetic reprogramming paints a really clear picture.
Right.
We are moving from basic life extension to precision, multi-layered biological engineering.
Which is truly staggering. But every engineer knows a system is useless if you can't measure its performance objectively.
Yeah.
And that brings us to the measurement revolution, driven by AI and biomarkers.
Right.
Why is the scientific community so unified on this? Why is it that without standardized biomarkers, the entire field just stalls?
It's purely logistical and economic. As we touched on, if you're testing a drug's effect on aging in humans, using lifespan as your endpoint is it's impossible. A multi-billion-dollar phase 3 clinical trial cannot last 50 years.
Of course not.
So validating any longevity intervention, whether it's D plus Q, rapamycin, an epigenetic pill, it's impossible without objective, quantifiable parameters that predict biological age and respond reliably over short, clinically realistic timeframes.
So biomarkers are basically scientific shortcuts. They allow clinical trials to be measured in months, not decades.
Exactly. They compress time and cost. The consensus from major global meetings like the Biomarkers of Aging Conference is unequivocal on this. We need biomarkers that are independent predictors of future mortality and disease risk, and they have to be highly responsive to interventions.
So what's the status of the leading biomarkers right now? Which ones are earning their validation stripes?
The most scientifically validated class of biomarkers right now are the DNA methylation clocks, often just called DNAMage.
DNAMage.
These clocks analyze the patterns of chemical tags or methyl groups on our DNA, and these patterns change in really predictable ways as we age, reflecting, cumulative damage and cellular dysfunction.
So they aren't looking at the genetic code itself, but the software layer on top of it, which genes are turned on or off, and that software layer changes as we get older.
That's a perfect analogy. The DNAMage clocks have now solidified their status as highly validated biomarkers because they can predict mortality risk and age-related disease onset independently of a person's chronological age.
So you might be 50 on the calendar, but the clock might say you're 65.
And that 65 is a significantly more accurate predictor of your risk of developing heart disease in the next 5 years than your actual birth date is.
Okay, that makes them powerful predictors.
Mm-hmm.
But how reliable are they at measuring change? Can we use them to prove an intervention actually works?
This is where the most compelling data point of the week comes in, the confirmation of their malleability. Our research team highlighted a rigorous, randomized controlled trial designed to test exactly that.
And the participants went through an intensive multimodal lifestyle intervention for, 8 weeks?
Just 8 weeks. And this wasn't just, eat your veggies. This was an aggressive, comprehensive intervention designed to hit multiple longevity pathways at once.
What did it include?
A specialized plant-rich diet, structured exercise regimens, specific sleep optimization protocols, and a defined set of evidence-based nutritional supplements. It was high commitment, designed to maximize biological response.
And what was that quantifiable response after just 8 weeks of that extreme program?
The results were statistically significant and really a landmark for the field. The participants saw an average 3.23-year decrease in their biological age.
Wait, say that again?
A 3.23-year decrease in biological age, as measured by the highly sensitive Horvath DNAMage clock.
8 weeks of focused effort shaved over 3 years off their biological age.
That's
staggering proof of concept.
It proves two absolutely vital things for geroscience translation. First, that biological age is not only reversible, but it is rapidly reversible.
Right.
And second, and this is crucial for drug development, it validates that these advanced epigenetic clocks are sensitive enough to accurately quantify therapeutic effects over very short clinical timeframes.
It gives the field the objective endpoint it needs.
Exactly. Now we can take interventions like D plus Q and rapamycin into large-scale trials and get a result in months.
But I have to ask the critical question here. For the average listener, is an 8-week, multimodal, high-intensity intervention really sustainable? What do the sources say about keeping those 3 years of reversal?
And that's the challenge. The study proved malleability, not sustainability. Experts absolutely acknowledge that compliance and long-term adherence to programs that rigorous are low.
So what's the point?
The goal of this data isn't to prescribe a crushing lifestyle change. It's to demonstrate that the body's aging trajectory is plastic and, crucially, measurable. This opens the door for pharmaceutical interventions to achieve similar, measurable results with a far lower compliance burden.
So the data validates the tool, which paves the way for the drug discovery process.
Precisely.
And speaking of accelerating drug discovery, the final element here is artificial intelligence. AI isn't just a computational aid anymore. Our research suggests it is fundamentally changing the speed of scientific discovery. How dramatic is this compression?
It's exponential. Our analysis indicates that AI systems are now capable of compressing research cycles that once spanned years into mere days. This is done through ultra-rapid virtual screening and analysis.
What kind of scale are we talking about?
The research team emphasized the sheer scale that AI offers chemical discovery. It can achieve a throughput capable of processing trillions of molecules in a virtual environment.
Trillions of molecules. That's a chemical universe that no team of humans could ever screen manually. AI is finding the needles in the haystack of potential longevity compounds almost instantly.
It is. But the power isn't just in finding new drugs, it's in fixing old clinical trials. The sources highlighted this incredibly innovative application of AI in clinical data analysis that promises to significantly boost the success rate of therapeutic development.
We mentioned the high failure rate in fields like Alzheimer's research. How did AI step in to fix that problem?
So AI was successfully deployed to rescreen data from a previous Alzheimer's trial that was initially declared a failure.
The traditional analysis showed no effect.
Right, no statistical difference between the drug and placebo groups.
Yeah.
The problem with Alzheimer's is its heterogeneity. Some patients progress rapidly, others slowly, and a drug might only work on a very specific molecular subset of people.
So the variability in the population just washes out the real signal.
Precisely. The AI algorithm went beyond simple averages and stratified the patients into these ultra-precise subgroups based on their molecular signatures and progression rates.
So it could separate, say, rapid progressors with high inflammation from slow progressors with low amyloid.
Exactly. And by isolating the patients most likely to respond to the drug's mechanism, the AI revealed a robust, statistically significant drug effect that had been completely hidden in the noise of the overall population.
That is remarkable. So the drug wasn't bad. The trial design was just too blunt. It's the difference between treating a whole forest with one approach versus treating only the specific trees that need that fertilizer.
A perfect way to put it. And this capability is transformative. It means companies no longer have to abandon promising drugs because of a perceived failure. AI can identify precisely who benefits and then optimize future trials to target only those responders.
It saves immense time and money.
And it accelerates the path to market for targeted therapies in all these complex age-related diseases. If you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could leave us a rating and review for Tomorrow Unveiled. It truly helps us reach new listeners.
So we've covered astounding progress today, from chemical synergies to genetic engineering and the power of AI measurement. But now we have to pivot to the biggest roadblock, the structural challenge that prevents these breakthroughs from reaching the public.
Right.
What is the single biggest impediment to widespread health span extension right now?
It is, without a doubt, the financial and regulatory bottleneck. You could call it the regulatory economic paradox. The science is ready, the targets are proven, but the path from a small pilot study to an accessible public health tool is almost entirely blocked by the economics of pharmaceutical development.
Okay, so let's use the example we started with. Dasapin and quercetin, or D plus Q. We saw encouraging cognitive benefits in that pilot study. Why can't a company just jump in and fund the necessary large-scale phase 3 trial to prove it works?
Because D plus Q are generic, inexpensive compounds. They are potentially highly effective, highly cost-effective public health tools, but they lack patent protection.
And large-scale phase 3 trial costs.
What? Hundreds of millions of dollars, often north of 300 million.
And a pharmaceutical company invests that enormous sum because, upon approval, the patent gives them a monopoly to sell the drug at a high price for years, which is how they recoup their investment.
Exactly. And without that patent protection, there is no financial incentive, no mechanism for any organization, private or otherwise, to fund those massive confirmatory trials. The return on investment simply isn't there.
So these high-value, low-cost, generic longevity compounds just languish in pilot study purgatory indefinitely.
That's what happens. The system is structurally biased toward expensive, patentable, novel drugs, even if the generic, affordable solution is biologically superior or safer.
That is the core structural flaw that leading geroscience policymakers are pointing to. This regulatory economic structure ultimately obstructs the path to accessible, cost-effective public health gains.
And it creates a massive societal cost. I mean, if D plus Q works to slow Alzheimer's, but we can't prove it because no one will pay for the trial, think of the lost health span years, the patient suffering, the healthcare expenses.
So what are the solutions being discussed at the policy level? If traditional pharma won't pay, who should?
The conversation is converging on the idea that these types of trials have to be treated as a public health imperative, kind of like funding basic research or vaccine development.
So new funding models.
Yes, policy experts are advocating for alternative models. Things like public-private partnerships, specialized philanthropic funds focused entirely on generics, or a market entry prize system.
What's that?
That's where the government offers a substantial multi-hundred-million-dollar prize for successfully completing the trial and getting an approved indication for a generic compound like D plus Q.
That seems logical. You reward the successful completion of the trial, not the patent exclusivity. It completely flips the financial incentive.
And it provides the necessary funding without restricting access to a low-cost drug. The consensus is that until a policy solution like this is adopted, we're going to keep seeing incredible scientific progress that never translates into widespread, affordable public benefit.
Beyond the funding, what about the regulations themselves?
Yeah.
We know the FDA regulates diseases, not aging.
Well, the field recognizes that before any major regulatory shift can happen, it has to speak with a single, unified voice. So there's a universal need for standardization.
Defining terms.
Exactly. Experts are calling to define and harmonize core terminology terms like longevity, health span, geroscience, to create coherence in research and policy engagement around the world. If the scientific community can't agree on what it's measuring, regulatory bodies will never trust it.
And if they do achieve that standardization, what's the biggest ask of regulators like the FDA?
The biggest ask is to establish approved clinical endpoints that measure improvements in functional longevity or the contraction of multimorbidity.
Meaning, moving beyond the current single disease model.
Exactly. Instead of approving a drug only for treating arthritis or only for diabetes, they want the FDA to approve a drug for the indication, slowing the incidence of age-related diseases, or extending functional health span.
This is the whole point of the TAME trial testing metformin against aging. That trial is designed to prove that by intervening in the aging process, you delay the onset of a cluster of diseases, not just one.
Precisely. And the success of trials like TAME is critical for validating these geroscience endpoints. If regulatory bodies can be convinced that slowing aging is a valid, measurable, and treatable indication, it unlocks the entire investment community. It creates a massive unified market for health span extension.
And finally, we have just unprecedented amounts of data being generated, genomics, proteomics, patient records. How are leaders coordinating all of this information?
The final imperative is coordinated global research and data sharing. The leaders in the field recognize that aging biology is far too complex to be solved in national or institutional silos.
Right.
So they're calling for coordinated global initiatives to connect disparate data sets, all this multi-omics and longitudinal cohort data, and then leveraging AI and machine learning to maximize discovery.
Learning from other fields.
Yes. Actively emulating best practices from other complex data-heavy fields like astronomy or climate science to create a truly global data infrastructure for longevity research.
So it really sounds like the science is now ahead of the policy and the economics. The breakthroughs are here, but the delivery mechanism to the public is still stuck in a 20th century regulatory model.
That is the tension that defines the field right now. We have the tools, we have the measurements, we have the engineering capability, but we lack the streamlined policy pathway to make it accessible to everyone who could benefit.
This deep dive on Tomorrow Unveiled has mapped out an extraordinary week. Let's just quickly recap the confirmed shifts.
We confirm that chemical interventions are moving towards safety and synergy. Metformin is proving its immense value as a chaperone drug, mitigating the side effects of powerful drivers like rapamycin and making them clinically viable.
And Senolytics, like D plus Q, are getting that crucial initial validation in humans. We have a direct, measurable finding that clearing senescent cells reduces inflammatory markers like TNF-alpha, which correlates directly with improvements in cognitive health.
It really solidifies the Geroscience Hypothesis in human data.
It does. Then on the engineering front, we've broken a decades-long technical barrier with the successful editing of mitochondrial DNA in human cells. That provides the final necessary pathway for cellular energy optimization.
And critically, those expert projections are accelerating. They're anticipating pharmacological age-reversal pills could be ready for human use within the next decade, thanks to controlled partial reprogramming.
And driving all of this is the measurement revolution. AI can screen trillions of molecules and salvage failed clinical trials by pinpointing specific responders, which ushers in an era of precision longevity medicine.
And meanwhile, epigenetic clocks have moved past just prediction. They now confirm malleability, reliably measuring a biological age reversal of over 3 years in a mere 8-week intervention.
The entire shift, it seems, is away from extending mere total lifespan, the quantity of years,
to maximizing functional health span, the quality of those years. The core scientific mission is now an engineering problem, ensuring you spend the maximum amount of time in good health and high vitality.
And as we close this, we have to remember the policy bottleneck. The most profound breakthroughs might be the low-cost, generic ones, but our system just isn't built to fund their validation yet.
We love hearing your thoughts. Whether you're on YouTube or your favorite podcast app, leave a comment and let us know what you think about this ethical and policy challenge of proving these effective, low-cost generic drugs.
And finally, while we track these aggressive timelines for chemical and genetic engineering, we should never forget that some of the most powerful interventions are non-pharmacological and accessible to us right now.
Right. Our sources close with a fascinating, actionable insight. A large-scale study of 86,000 people showed that speaking more than one language, multilingualism, was found to have the risk of accelerated cognitive aging.
Have the risk.
Have it. And this potent finding reminds us that cognitive reserve, which you build through challenging, lifelong learning and complex mental activity, it remains a powerful, accessible tool we all have today for preserving functional longevity, regardless of what future drug breakthroughs come along.
A profound and actionable reminder to end on. Thank you for joining us on Tomorrow Unveiled.
We'll be back next week to continue mapping the path to a longer, healthier future.